All right, guys, so we're going to start looking at cells today. And I know that you have done cells probably last year, and I believe you did them again in 7th grade. So this was like your 3rd time going through cells. So it's not anything new to you. And I'm not going to spend a whole lot of time harping on them. I just want to go through and tell you the basic structure and function, make sure you can recognize them, know who important people were. Okay, well, now I'm starting to go on, and that sounds like a lot. So let's just get into it then. Overall, our cell unit is normally pretty quick. And it's not a whole lot of me talking, it's more you guys doing things. So we're going to look at the following pictures, and we're going to classify them as either plants, animal, or bacteria, and we're going to discuss what would lead you to this conclusion. Now, if you were in class, I would go through these slides, and I would pick on random people to answer these questions. Since it's online, I'm not going to go through the entire PowerPoint because it's just the same couple cells over and over and over again. So look at this one here. You'll notice it has a boxy structure on the outside, and it has a huge vacuole in the middle, and a cell wall is labeled. Knowing what you know about cells, hopefully that makes you think that it is a plant cell. If the vacuole in the cell wall didn't give it away, there are chloroplasts in there as well, right here. That's what makes it green. So whenever you're trying to identify a plant cell, you want to look for a large vacuole, a cell wall, and a chloroplast. Looking at this guy now, he is a lot smaller. He does have a cell wall, but I don't see any chloroplast in there, and he has these cute little flagella. Some cells have them, some cells don't. They're used for movement, mainly. This is going to be a bacteria cell. Notice how much smaller it is and how much more simple it appears. All right, looking at this one here. Again, it has that boxy structure. I see some chloroplast labeled, and I see a cell wall labeled. Automatically, I'm going to go with a plant cell. Here's one we haven't seen yet. There's only one type of cell left. There is no boxy structure. I don't see any chloroplasts that are labeled. This one is going to be an animal cell. It is normally circular or oval in shape. Sometimes it looks a little wonky, but it does not have that box shape, and that's what we're looking for with an animal cell. All right, looking at this one, again, I see chloroplast, and we have a nice little square, so this one is definitely going to be a plant cell again. Even though nothing is actually labeled on here, I can see that flagella. I see the DNA material is just kind of chilling inside the cell right here. That is going to be a bacteria cell again. All right, we have a circular shaped cell. Since it's not a box, that's going to be an animal cell. Are you guys starting to pick up on how to do this? I hope so. Cell wall is labeled here. Got some chloroplast labeled. It is boxy in shape. Now it's more of like what we call that, like an octagon, but it's still boxy. It has specific corners. That's going to be a plant cell. And here we have another bacteria cell. They all look like little pill capsules, which is kind of ironic to me because whenever you get sick, you end up taking a pill to get rid of the bacteria. And bacteria are kind of shaped like little pills. At least these ones are. All right, so I'm going to stop there with going through all these cells. And I'm going to get back into the actual lesson here. Let's see. We don't need that one anymore. We'll go with cell structure and function. Now, we're going to be doing this in class on Monday and on Thursday. Tomorrow we have an Act 80 days, so you guys will not actually have class, and I labeled that for you in Canvas already. So this video will be posted for you again on Thursday's class, same exact video. So the first part of the cell that we want to talk about is the cell membrane, and this is the most outer layer of a cell. It's a semi-permeable membrane, which means that it allows certain things in and certain things out, but not everything. And it's going to surround the cytoplasm. Its main job is to keep some things in and some things out. So this is what it looks like. You have 2 layers of phospholipids. This layer is going to touch the outside. This layer is going to touch the inside. And it has these little carbohydrates attached to it. And that is going to be like little signals for the cell. It's how it talks to other cells and communicates its needs. Now, this big thing here is a protein. It's called an integral protein. It's that big pink thing, this guy right here. And he is going to be allowing those carbohydrates in and out because they cannot pass through the cell membrane itself. Cholesterol is a hormone. Hormones, this little guy here, they can go right through the membrane because they are considered polar. And that is the quickest way for cells to signal, actually, using a hormone. I talk all about that in my genetics class, so if you have any questions on cell signaling, find one of them to ask about it. The cytoplasm is a jelly-like substance within the cell. It's just outside the nucleus, and this is what all the organelles hang out in. So if you experience a little bit of trauma, the cell parts shouldn't be damaged because that cytoplasm is just going to wiggle and jiggle like jello and keep everything nice and intact. Nucleus is the central, most important part of the cell, and it's going to guide the activity and the growth. And here are the different parts of the nucleus. It's kind of like a mini cell in itself. It has little pores that allow things to go in and out of the nucleus. Yes, things do leave the nucleus. It has a nuclear envelope, which is similar to the cell wall or the cell membrane in our cells. The nucleolus is that very dark part in the middle there. And then the chromatin or the chromatin, depending on how you want to pronounce it, that's your DNA before it begins to divide. Endoplasmic reticulum is a network of tubes involved in protein and lipid synthesis. That means that they make proteins and lipids. You can always tell the rough ER from the smooth ER because the rough ER has all these little dots, and those little dots are ribosomes. Ribosomes are like little packaging centers. So they assist in making these proteins and lipids. The smooth ER does not have them. So I have that picture over here. This is what the smooth one looks like, and there's your rough one. The Golgi apparatus is packaging and transport. They so lovingly refer to him as a mail carrier. Lysosomes contain digestive enzymes, and your white blood cells are actually lysosomes. So they go around, they clean the cell up. If there is an old, worn-out organelle, they'll eat it, and they also eat bacteria cells. So lysosomes are good. A chloroplast, these are going to absorb sunlight and use it to produce food in autotrophic organisms. Auto means self, so they feed themselves, and they are green. We'll talk a lot about the chloroplast when we get to chapter 6, I believe. We'll actually go in detail about the granum and the thylakoids, which are these little guys here that are inside of the chloroplast. A cytoskeleton, these are microtubules and microfilaments that help organize and give the cell its shape. So you got your cell wall, not your cell wall, you have your cell membrane here, and we got a protein here, and then these blue things coming off, those are going to be the cytoskeleton. And it's very similar to your skeleton in your bones. Now you don't think of, breaking a cell, but it just helps to keep the cell membrane from, completely collapsing in on itself. A vacuole is used for storage or for water, waste, and food particles in larger plant cells. Now, we have a vacuole as well. It does not function the same way that it does in a plant cell because plants mainly store water in their vacuoles, but we do the other things. We'll store waste or food in it until we can get rid of it. In a plant cell, though, the vacuole is going to be like the biggest organelle. A cell wall is a rigid layer of polysaccharides lying outside the plasma membrane. Plasma membrane is another word for cell membrane, and that's what gives it that boxy structure. When we talked about density and water freezing in chapter 2, I mentioned frozen vegetables, and I said that whenever water freezes, it actually expands, and when it expands inside of a plant cell, it pops their cell wall and it cracks it, which makes them not so crunchy anymore. A thylakoid is a flattened sac inside a chloroplast. It is the site of photosynthesis. So there are all the specific parts of a chloroplast. Know what a thylakoid is? I'm not going to go into all those right now unless we see a few more popping up here. Well, nuclear membrane. This is a membrane surrounding the nucleolus. So that nucleolus is the purple part there, and this whole thing is the nucleus. Now, the nucleolus is a small, dense sphere located in the nucleus. Ribosomes are going to consist of RNA. It's a site of protein synthesis. Flagella are whip-like tails that allows the cell to swim. And cells can have no flagella, 1, 2, 4. They can have a ton of flagella. I don't really particularly like how this one looks. It reminds me of a spider, and I don't enjoy that. Pili, similar to a flagella, they're hair-like structures that are also used for movement. Think of like a thousand-legger. Pili are going to be all around the outside of the cell. So flagella normally come off of one end. Pili are much smaller than the flagella. Mitochondria are going to produce ATP. You know, the mitochondria is the powerhouse of the cell. I don't know how many times you guys have heard that one before. Chromosome is a structure of nucleic acids and proteins, and it carries genetic information, which is your DNA. In bacteria cells, it is a nice little circle. In our cells, it forms an X. Differences between plant and animal cells. To begin with, plants have a cell wall, a chloroplast, large vacuole. Generally, they are larger in size and they have a fixed shape. They're always going to be that square, rectangle, boxy looking shape. Animals. We have lysosomes, plants don't. They are generally smaller in size and they are irregular in shape, meaning that they can be a circle, a little oblong, a little wonky looking. They don't necessarily have to be a perfect circle. And they normally do not have corners. Similarities, they are both eukaryotic, and they have all other membrane-bound organelles in common. When we talk about prokaryotic versus eukaryotic, I'm talking about bacteria cells, which are the prokaryotes. Pro means before, and karyon means nucleus or membrane-bound organelles. So a prokaryotic is before membrane-bound organelles. Eukaryotic cells are like us and plants. The prefix "u" is going to mean with, and karyon is nucleus, so that is with a nucleus. So prokaryotes, they do not have membrane-bound organelles. Remember, pro means no. They have nothing inside of them like that. They are the bacteria and the archaea. Usually they are unicellular. They have a plasmid, and plasmids are pretty cool because it gives that bacteria cell some type of a benefit. And they are in size 1 to 10 UMs, which is extremely, extremely, extremely little. Tiny micrometers. Eukaryotes. They contain membrane-bound organelles. These are going to be plants and animals. Usually they are multicellular. Now an amoeba is unicellular and they are eukaryotic. There are always, those random ones that don't exactly fit the form. They have chromosomes in the shape of an X instead of just a plasmid, and they are bigger, 10 to 100 micrometers. When I say bigger, you still can't see them.
Please tell 5305. This is C-Rock 5306. I'm sorry.
Moving on, important people, there are 6 of them. The first one is Robert Hooke. He examined cork under the microscope and he named the boxes cells. He said it reminded him of being in a monastery and the monks all lived in cells. You guys thought I was going to say prison, didn't you? Antoine van Leeuwenhoek is the second guy. He observed pond water and he discovered protists. He really discovered microorganisms. Theodore Schwann identified the cell as a basic structure of plant and animal tissue. So he went out and said that animals are made of cells. And I always remember him because Schwann kind of sounds like swan, and swans are animals. Matthias Schleiden, he determined that plants are composed of cells. Rudolf Virchow stated that all living cells come from other living cells, so he started us off with the cell theory. And Louis Pasteur, good old Louis, he disproved spontaneous generation. He basically said that you can't have something come from absolutely nothing. There has to be some type of a catalyst to get it going. The cell theory comes in 3 parts. The first part is that all organisms are made of 1 or more cells. The second part is that cells are the basic unit of life. And the third is that cells arise from pre-existing cells. So they come from other cells. And that is it for the cell notes as far as we're going to do with those. For the rest of it, you guys are going to be doing little activities, and it's going to really end with you guys doing this big SpongeBob project next week. Alrighty, well, that is all I have for you today. So I will see you guys later.